Starting off with enzymes, which is the second part of the chapter biological molecules. Enzymes are proteins. They are biological catalyst. Catalyst is anything that speeds up the reaction. And biological because they are produced in the body. They are made in living things and they speed up the rate of chemical reactions. They are all specific to one particular substrate as the active site of the enzyme, where the substrate attaches is complementary shape to the substrate. For example, proteins would only fit into proteases, carbohydrates would only fit into their enzymes, carbohydrates, lipids would fit into lipases. So they are all having a complementary shape. If the enzyme is like this, then the substrate would have an exact complementary shape in order to fit inside this puzzle. Only then a substrate enzyme complex would be formed that we call as this. This is known as a substrate enzyme substrate complex, ES complex. Now, enzymes are affected by changes in temperature and pH as well. And also, you should know they can be reused at the end of the reaction as well. They are not used up in the reaction, they catalyzed and they can be reused over and over again as well. Now, here you can see this substrate is specific only to enzyme number 1. It cannot fit in enzyme number 2 because the shape can't fit over here. So enzyme 1 is complementary in shape to the substrate and enzyme 2 is not able to bind to this substrate because they are not complementary. Substrate and enzyme join like a lock and key. Here you can see when they once form this lock and key ke baad, lock and key is a hypothesis. And once they form this enzyme substrate complex, now the enzyme is going to speed up the reaction by breaking the substrate into two parts. And the enzyme you can see it's the same form that it
Still.
was in the start of the reaction, and at the end of the reaction, it's still the same. Got it?
Yeah.
Unchanged. Enzymes are proteins and they also have a specific shape held in place by the bonds. Now, this is extremely important around the active site area, and the specific shape is what ensures the substrate which will fit into the active site and enable the reaction to proceed. Enzymes work
Yes.
the fastest at their optimum temperature in the human body, and in the optimum temperature, human body में क्या है? It is 37 degrees Celsius. Heating
Yes.
to high temperatures beyond the optimum will break the bond that hold the enzyme together and it will lose its shape. This is known as denaturation, where the substrates cannot fit into the denatured enzyme as the shape of their active site has been lost. When you increase the temperature, starting में क्या होता है? Starting में the reaction would increase. Why? Because temperature is increasing. Kinetic energy of the molecules is also increasing. They are moving faster. Good reaction. But only to a certain temperature. Increasing temperature is good. It increases the rate of reaction, but to one extent, when it crosses the optimum temperature, then the enzyme is going to denature. Optimum temperature is the best temperature where the enzyme is working exceptionally. Denaturation is irreversible. Once the enzymes are denatured, they cannot regain their proper shape, and remember, their activity will stop. Increasing the temperature from 0 degrees Celsius to optimum increases the activity of the enzymes as well. As more energy the molecules have, the faster they will move, and the number of collisions with the substrate molecule would also increase, leading to a faster rate of reaction. This means that low temperatures do not denature enzyme, they just make them work more slowly. Got it?
Right. Yeah.
So here you can see, can you tell me what has happened when the temperature was increased?
They become denatured and won't fit into the lock and key mechanism.
You can see the shape has changed of the
Mm-hmm.
enzyme. Now this substrate can no longer fit in the enzyme and the substrate won't be breaking. If the substrate won't break, it would be difficult now for the substrate to react as well. So when you
Yep.
start increasing the temperature, the enzyme become active. Good. It's very good to increase the temperature. But when it crosses the point, which is known as optimum temperature, then the enzymes are going to denature. For example, your body enzymes all are optimum to 37 degrees Celsius because this is your body temperatures. So you can see as we start increasing the temperature, more kinetic energy and the rate of reaction increases. This is where this is the optimum temperature, and this is the point where the enzyme starts to denature. And you can see it by the gradient of the graph that the rate is actually decreasing. So this is a picture of a graph showing the effect of temperature on the rate of an enzyme controlled reaction. Got it?
Yep.
So now we are going to perform a practical investigation. Okay, this is important for your paper number 2, where we are going to investigate the effect of temperature on starch solution as well. Let me show you one thing. So can you describe me what is enzymes and what are the different factors that affect the enzyme activity?
Enzyme, they are proteins, and they are biological catalysts which heat up the effect,
Good.
and they are affected by temperature. So high temperature would increase reaction, and low temperature would decrease it. But if you go over the optimum temperature, it would denature and won't fit.
Good job.
And this allows the enzymes to be really specific. In this video, we're going to explore the world of enzymes. So we'll take a look at what enzymes do and how they work, and then at the differences between the two main models of enzyme action, namely the lock and key model and the induced fit model. Before we get to enzymes themselves though, we first of all need to understand why enzymes are so important to us. The key reason is that in order for a living cell to function properly, it has to carry out a huge number of chemical reactions every second of every day. But the problem is that most of these chemical reactions are naturally quite slow. The most obvious way to increase the rate of a reaction would be to increase the temperature. But this runs into a few problems and makes it unfeasible. For example, it can require a huge amount of energy to keep an entire organism very warm. And those high temperatures can also damage our cells. Most importantly though, high temperatures would also speed up non-useful reactions that we don't want to happen. So because of all these problems, a better way to increase the speed of chemical reactions is with a catalyst. We can define a catalyst as a substance that increases the speed of a chemical reaction without being changed or used up in the process. And this last bit of the definition is really important, because it means that catalysts can be used over and over again to catalyze multiple reactions without being used up. Now, the word catalyst is a general term that you might see in chemistry as well. But an enzyme is a particular type of catalyst that's made by living organisms, and so we sometimes call them biological catalysts. Enzymes themselves are basically just large proteins. So, like all proteins, they're made up of long chains of amino acids. And depending on the specific sequence of amino acids in the chain, they can fold up to form different shapes. And each of these shapes would be a unique enzyme that catalyzes a particular chemical reaction.
Got it? What are enzymes?
They are proteins which are biological catalysts.
Good. So now we are going to heat up a starch solution to a set temperature, and iodine is going to be added to the wells of a spotting tile. We are going to take a spotting tile and we are going to add drops of iodine. Can you tell me what is the color of iodine?
Iodine is like reddish.
Orange. Okay, orange brown. Iodine is what? It's orange brown.
Brown, no, orange brown.
Okay, so I have added different drops of iodine on a spotting tile and I have started to heat up a starch solution. Now, in that starch solution, as it was being heated up, I have added the enzyme amylase. Can you tell me what does amylase do? It breaks down.
Okay, breaks down here.
Now, amylase is present in our salivary glands, which is responsible for breaking down starch.
Breaking down the starch.
Amylase is added to the starch solution and mixed well. Every minute, the droplets of the solution are added to a new well of iodine. Now, remember, there's a test for starch. Can you tell me what was the chemical test for starch?
You heat it first and then add iodine and wait.
You haven't revised the PowerPoints. Okay. Starch का test है. We add iodine solution. We don't heat, we don't do anything. We just add iodine and it turns blue-black. Got it? Remember now?
Blue black, yes.
It turns blue black. Simple as that. Okay, that was the test for starch. So I am going to add amylase to starch. Amylase is the enzyme and starch is the substrate in a jar, and I'm going to start heating it. Okay, and after every minute, I'm going to add a drop. Okay, now this solution is being heated. This solution contains starch as well plus the amylase as well. The solution is being heated. I'm going to take a drop from here after every minute and add it to the iodine. If the iodine turns blue black, what does it represent?
That the starch is present.
There is starch. Okay, after 2 minutes, I again added a drop. Starch is still present. 3 minutes, I still added. Starch is still present. But 4 minutes, when I added the drop, it remained orange brown. The color didn't change. What does that mean?
That starch is not
Starch is no
present.
more in the solution. The amylase has broken down all of the starch that was present. Got it?
Yep.
Amylase has done its job. This is continued until the iodine stops turning blue-black, which means that there is no more starch left in the solution as the amylase has broken all of it down. Now the time taken for the reaction to be completed is then recorded and the experiment is repeated at different temperatures. The quicker the reaction is completed, the faster is the enzyme working. Got it?
You're
So
right.
again, you add solution to a beaker, you start heating the solution, you have added a thermometer over here as well, and then you are going to use a spotting tile to add the drops of iodine onto the tile. Now what you're going to do, you're going to add amylase enzyme as well. And right now the starch solution is having a temperature of what? 30 degrees Celsius. Now you're going to
Right.
add amylase to starch solution and start the timer right away. After every minute, now you can see this experiment was done at three temperatures. Temperature 1, temperature 2, temperature 3. Okay. Now, if we look at what happened at 9 minutes, okay, what happened at 20 degrees Celsius, it took 9 minutes for the reaction to complete. 1, 2,
Right.
3, 4, 5, 6, 7. After ninth minute, with the iodine stopped changing color. So this proves that when you use a lower temperature, the enzyme activity is also slower and the starch breaks down late. Got it?
Yep.
But at 40 degrees Celsius, what happened? Can you tell me?
Uh, it only lasted from 20 or 1 minute,
What
and
lasted?
then the iodine only went to 3.
The starch. The starch only went till 3. The starch was now all broken down after the second minute. Got it?
Yes.
Iodine color remains the same. Iodine को कुछ नहीं हो रहा. ठीक है? Iodine is just the reagent.
Yes.
And at 60 degrees, why is starch present in all of these drops? Why hasn't it been broken down? You are going to tell me the answer.
Because it is in its optimum temperature.
Optimal temperature is a good temperature, which is 40. This is a good temperature that the reaction is only complete in 3 minutes. यहाँ पे reaction complete ही नहीं हो रहा. The starch is still present even after 15 minutes.
No. It enzyme has denatured, so
Exactly. Good job. Enzyme has denatured. That
denatured.
is why the starch is not being broken down because amylase is dead. Amylase is dead. Starch is always gonna be
Yep.
present whenever you add iodine because there is no one now over there in the beaker to break down the starch. Now we're
Yep.
going to talk about the effect of pH on enzyme activity. The optimum pH for most enzymes is 7, but some that are produced in acidic conditions such as the stomach have a lower optimum pH of 2, and some that are produced in alkaline conditions such as duodenum, they have a higher optimum pH as well, such as 8 or 9. If the pH is too high or too low, the bonds that hold the amino acid chain together to make up the protein can also be destroyed. This will change the shape of the active site and so the substrate will again no longer fit in. We'll say कि अगर यहाँ भी beyond the optimum temperature अगर बहुत ज़्यादा pH low या pH high change हो जाता है, the enzyme will again denature and the activity might stop again. Here you can see this is
Yep.
an active site. The substrate is right now able to shift, fit into the active site. But when you have pH is increased above the optimum one, the shape has changed and they can now no longer bind. Got it?
Yep.
Good. Now look at this graph and then you're going to explain me what is happening.
So the optimum pH is where it work highest best. So, but after it decreases because the enzyme has denatured and won't work and won't fit again. Hello?
Okay, and what has happened beyond optimum pH?
The enzyme has denatured, so they won't fit again, so it won't work.
Exactly. So now we are going to involve an experiment for a practical again to now demonstrate the effect of pH. Place single drops of iodine solution in rows on the tile. Label a test tube with the pH to be tested. Use the syringe to place 2 centimeter cube of amylase in the test tube and then add 1 centimeter cube of buffer solution to the test tube using a syringe. Use another test tube to add 2 centimeter cube of starch solution to the amylase and buffer solution and then start the stopwatch whilst mixing using a pipette. Now what we're going to do, we are going to label the test tube which we are going to test. Okay, and we are going to again add some amylase into the test tube as well, but at different pH. And now I am going to have a test tube of different pH. After 10 seconds, use the pipette to place 1 drop of mixture on the first drop of iodine, which should now turn blue-black in color. And then wait another 10 seconds and place another drop of mixture on the second drop of iodine. Repeat the same method every 10 seconds until all the iodine solution remains orange-brown, and you have to do this at different pH values. So this was the starch solution. This is the amylase solution, which is the enzyme, and you have added 1 centimeter cube of pH 7 buffer. In another test tube, you can use a different pH, and you are going to have a lot of different pH tubes. Got it? ये होगी
Right.
pH 7, ये आप कर लो at pH 14, ये आप कर लो at pH 2. And then you are again going to use the same spotting tile experiment. After every 1 minute when you start the stopwatch, you have to add 1 drop over there and you have to see where does the fastest reaction occur. Let's say the fastest reaction occur at pH 2, which means that the enzyme's optimum pH is 2. Why? Because it worked the fastest. It took only 2 minutes for the starch to disappear, which means the enzyme has had broken down all of the starch in only 2 minutes. Got it?
Yes, because that was the best temperature.
Exactly. But if you look at pH 14, there was no reaction. This means that pH 14 denatured the enzyme, and the enzyme
Yep.
was never able to break down the starch. Starch was present
Yep.
all the time over here. No starch was broken down. And that is the end of your chapter of enzymes. Got it?